Okay, welcome back. Let's talk about DNA replication, or how DNA copies itself. So, we are going to focus on bacterial cells when we're talking about this, but there's a lot of consistency between how bacterial cells do it and how other cells. So, the big picture is the same for other cell types. So, in DNA replication, we have these two strands. And you already know about base pairing. So when these two strands open up, one strand can act as a template for the second strand. So you see here, this is a double-stranded DNA, and it's opening up here. I'm at the replication fork. And where there's a T, it's adding an A. Where there's a G, it's adding a C. Where there's an A, it's adding a T. Same on this one. T, where there's an A, add a T. Where there's a C, add a G. There's a T, add a A. Right? So it's just base pairing. G and C go together, A and T go together. So what's happening first is that helicase is an enzyme that's going to separate these two strands together, or separate them apart from each other. While it's doing that, if you've ever had like something braided and you try to just pull it apart in the middle, it might kind of tangle on at the ends. We don't want our bacterial DNA to get broken. So these enzymes, topoisomerase and gyrase, are going to help relax those strands so that it's not breaking or tangling. It might help to wind it or unwind it to keep it relaxed. Gyrase here, I put a little asterisk after it. Gyrase is an enzyme that's pretty specific to bacteria. You typically only see it in bacterial cells. If we're looking at archaea, they have something called reverse gyrase, which is also able to help relax the strands. Although archaea use reverse gyrase more to keep their DNA from denaturing when it's really hot. So, but there's, okay, interesting anyway. Okay, so topoisomerase, gyrase, and helicase all together are working together to help create this replication fork. Helicase separating the strands, topoisomerase and gyrase stopping it from turning into a big tangled mess. So here's our two strands. Helicase, opening it up. And then what's going to happen next is we're going to add a primer. See, we're adding a primer and then start building DNA. You can see on this strand, it's facing towards where helicase is opening up. So once that DNA polymerase starts adding DNA, it just is going to keep going on and on and on forever. You have this nice continuous strand. That's the leading strand. But if you remember, our two strands are facing opposite directions. They are antiparallel. So this other strand cannot move towards the replication. It is going to move backwards away from it. So it'll put on a primer and go back and add it. Then it has to come play catch up. Put on another primer, start adding DNA. Put our primer, start adding a piece of DNA. These little pieces of DNA we get, those are called Okazaki fragments. And then those Okazaki fragments have to be combined. The primer has to be taken out and replaced with DNA, and the Okazaki fragments have to be combined to make one long piece of DNA. So that's DNA replication in one little simple cartoon. Okay, so some terms to make sure you're comfortable with. Replication fork, where replication is occurring. We just described that. Leading strand and lagging strand. We just described that. Leading strand made continuously, one long strand. Lagging strand made discontinuously. So it's made in different pieces, made in those Okazaki fragments. Then semiconservative replication. I haven't used that phrase yet. Semiconservative replication means that at the end of DNA replication, each new daughter cell is going to have a piece of DNA where one strand is from the parent and one strand is brand new made. So that's what we're seeing here. The two strands open up. So this cell has one black strand and one purple strand. This cell will get one black strand and one purple strand. That's semi-conservative. So the newly unwound single strand is going to combine with new nucleotides. So you have an original strand and a newly made strand that rewind together to make the new DNA. Okay, so here's another picture to look at it. Here is the replication fork. Vocabulary word to remember there. Make sure you can describe that. Here, DNA is just being made in one long piece. Is that the leading strand or the lagging strand? moving towards the replication fork. That is the leading strand. Here we have DNA being made in little pieces. What are the names of those little fragments? Do you remember? Those are Okazaki fragments. And these pieces are being made discontinuously. So is this the leading strand or the lagging strand? That's the lagging strand, made in pieces, moving away from the replication fork. And as these enzymes move on down, then the next RNA primer will come over here, and you'll have a new primer made. So a few different terminology here. In that little cartoon, it called it primase. In your textbook, it calls it RNA polymerase. So this is the phrase that I will use. On quizzes or exams, you will say RNA polymerase. RNA polymerase is what puts the primer on. And that primer, as you may have guessed from the name, is made of RNA. So that RNA primer does have to be removed and replaced. DNA polymerase is going to remove the primer and put DNA in it. So that gives us two different kinds of DNA polymerase. We have DNA polymerase 3, okay, and DNA polymerase 1. DNA polymerase 3 is what is going to chug onto that primer and just start adding DNA to make all the DNA going on down. So this is DNA polymerase 3, adding DNA on top of the primer. DNA polymerase 1 is what cuts out that primer and puts DNA in there, so you don't have any RNA left. Okay, I just said a lot of big words. So if you need to go back, make sure that you can tell me what's the leading strand, what's the lagging strand, what's Okazaki fragment, what's a replication fork. You already can tell me helicase, gyrase, topoisomerase. On this one, you need to now be able to tell me RNA polymerase, DNA polymerase 1, DNA polymerase 3. Okay, so pause for a second, see if you can list what those things all are. What is a leading strand? What is a lagging strand? What is a replication fork? What is an Okazaki fragment? What does it mean to be antiparallel? What does it mean to be semiconservative? What is helicase doing? What is topoisomerase doing? What is gyrase doing? What is RNA polymerase doing? What is DNA polymerase 3 doing? What is DNA polymerase 1 doing? And at the end, what is DNA ligase doing? Okay, so jot down your answers to that. And then voila, here's some answers. DNA polymerase 3, did you say it's adding nucleotides onto the 3' end, onto the primer? Did you say RNA polymerase is adding nucleotides, is adding the primer? DNA polymerase 3 adds DNA to the primer. DNA polymerase 1 takes out the primer and puts in DNA. And DNA ligase will ligate or join those Okazaki fragments. Okay, any of those enzymes are up for the exam. I can give you any of them. Say, what is this doing? What is that doing? Okay, in most bacteria, DNA replication is going to be bidirectional. So let's actually look at a picture here. So here is where replication is starting. See, it's moving around in both directions. So we're having a replication fork go this way. We have a replication fork go that way. That's what it means bidirectional. Each offspring is going to have one copy of DNA, and replication is going to be very accurate. That's super important. We don't want to just wildly mutate, and who knows what happens to your offspring. We have very accurate DNA replication, and that's in part due to DNA polymerase, that while it's moving incredibly fast speeds, it stops and proofreads itself at every step to make sure that it's adding the right nucleotide. Okay, then at the end, after it's come, here's where it started. It's moved around in both directions. Here's termination of replication. So that site, it has gone all around the chromosome, and the two pieces are going to let go and go to two daughter cells. Okay, so what is DNA replication? What does that mean? What are the roles of these enzymes? Gyrase, ligase, DNA polymerase. From that, you should also be able to say DNA polymerase 3, DNA polymerase 1. A lot of terms I stopped and talked about. Make sure that you're comfortable with those words. Okay, I will see you back. We'll talk about transcription and translation in our next video. Bye.